Okay, well, thank you so much, Gary, for that introduction. I'm so happy to be here. This is a wonderful, I was looking at the booklet. It's a great meeting. I'm excited to talk and hear all about the work that's happening here. So as Gary said, my lab is a cancer neuroscience lab, and we are very interested in how nerves are recruited to tumors and what they're actually doing once they're there. And so to start with, I just wanted to give a background on the types of cancer that we work on, just so everyone's on the same page. So we work on predominantly head and neck cancer, though we have done some ovarian cancer work. And these are cancers that arise in the oral cavity. And there are two basic types. They're these umbrella terms. The biggest one, these are about 75% of cases, are mutationally driven. So those are, the risk factors are smoking cigarettes, drinking alcohol. These are typically older gentlemen that have this type of cancer, though women are starting to increase in this. The smaller 25% of cases are induced instead by infection with high-risk human papillomavirus, and those are termed HPV positive, obviously. We have lots of therapies that we can use to treat these patients. There's chemo, radiation therapy, targeted therapies for those that have upregulated, for example, EGFR. Immunotherapy now is another one that's a go-to, though not everybody responds as robustly as we would like, and obviously surgery. We can cure some of these patients, but inevitably those that we can't, recurrence and metastasis is a challenge. We don't do very well with these patients, and we need to do better. Given our interest in the nerves, we started wondering, well, are the nerves somehow involved in this recurrence and metastasis? If we can understand what they're doing, can we therapeutically target it and help these patients? So we started by looking at these two different types of cancers. No one had really asked the question, is there a difference in innervation of these tumors? So we partnered with Desiree Muirhead, who's the head of pathology at Stanford, and we stained HPV positive and HPV negative samples for beta-3 tubulin, which is our pan-neuronal marker. And we gave these slides to Des, and she was completely blinded to the HPV status. And we're like, here, score these for us. And we were really surprised. It was the HPV negatives that are really densely innervated. I actually had assumed they would be the same, and they're really not, which is interesting because they occur in the same region in the oral cavity. So why the difference, right? So this was our first step to start thinking, okay, the nerves are probably doing something here. In addition to our thought that they might be involved in recurrence and metastasis, we started to wonder, are they involved in the initiation of these cancers? Do we need to go earlier than metastasis and recurrence to look at their role? What we knew was that tumors release a lot of different things into the microenvironment. And this is not just our work. Many people have shown this. They release soluble factors, things like neurotrophic factors, NGF, axonal guidance molecules, and small extracellular vesicles, also known as exosomes. These are all released by the tumor. These factors diffuse local regionally, and they get received, these signals get received by local axons. These are coming from ganglia, predominantly the trigeminal. And these signals induce sprouting and innervation of the tumor. And these are guided by these guidance molecules. And we can do tricks in mice. And a lot of people do this in the field where you could either chemically ablate the infiltrating nerves. And in the case of head and neck cancer, these are sensory neurons. You can chemically ablate them. And what we see is the tumors grow slower, suggesting that the nerves are inducing some sort of proliferative response in the tumor. We can also delete them genetically, and we see the same thing. So they're doing something important, and we can control this in our animals. Of course, we're not going to necessarily ablate nerves in patients, so we have to understand more what they're doing to have targeted ways to address these nerves. But we went back to the exosomes and we wondered, okay, they're being released and they recruit the nerves, but what happens once the nerves are already there? Is there a dependence on these exosomes still in a different capacity for tumor growth? Are they involved in treatment resistance? Maybe exosome release stops once the nerves are there. And so it was a question we were very interested in. And a former very talented MD PhD student in my lab, Tony, who's now in the physician scientist program at Pitt, he said, all right, I'm just going to do this very simple study. We have an HPV positive line that Tony was working on. So we ended up looking at HPV positive for this study. And we implanted this Merle cells into wild type animals. We had a variant of Merle cells that were compromised in exosome release. We had CRISPR'd out RAB27A and B. So these are the parental Merle cells that now cannot release exosomes. And so he put these two tumor lines into wild type or ablated animals and simply tracked tumor growth. What he saw was what we had seen in the past. So here in the green, these are just wild type tumors in wild type animals. The tumors grow very nicely. If we put the exosome compromised tumors into wild type animals, we can decrease tumor growth, which we had seen before. The exosomes are recruiting the nerves. If you can't recruit the nerves, we see smaller tumors grow. Now, if you put the wild type tumors into the ablated animals, they grow slower. We knew that. But this was the surprise. When he put the exosome compromised tumors into ablated mice, no tumors grew. And I'll be honest, when he first showed me this, I was like, no way, do it again. And the same thing happened. And so then I believed him that this was actually really interesting. The exosomes are doing something very, very important for tumor initiation. So how are they doing this? And if they're affecting initiation, our next thought was they must be altering the neurons in some way. And so Tony said, all right, well, let's just look at that. He isolated trigeminal ganglia from the ipsilateral and the contralateral side of tumor-bearing mice, and then did some RNA-seq. And what he found was that on the ipsilateral side, there was a really high expression of ATF3. This is a master transcriptional regulator that's turned on in neurons when they're injured. And it sets up this program of injury repair, which induces expression of these, they're called RAGs, regeneration-associated genes. And those are really high on the ipsilateral side. There was also increased expression of CGRP, substance P, and beta-3 tubulin, which is our pan-neuronal marker. So the tumor was somehow, and the exosomes were somehow initiating these transcriptional changes on the neurons. Okay, so what does that mean? Does it affect the secretome of these cells? Tony really just having isolated the ganglia, he just stained for substance P, which is one of the major neuropeptides that's released by these sensory neurons. And he found that, yep, when he quantified substance P, it was really higher on the ipsilateral side. And then he did a cytokine array. So he took conditioned media from the tumor cells that were co-cultured with neurons and threw it on this array, and we saw differences. This was actually really unexpected. There are major differences in things other than the traditional neuropeptides that we had expected to see. And we really were really interested in IL-6. So that's a cytokine that does many, many different things for the immune response. And we know that the immune response in head and neck cancer and other cancers plays a critical role. And so we kind of honed in on IL-6. And we asked, okay, what are the next changes that we want to look at? What should we focus in on? And we started looking at the effect of the neurons on neuropeptides. So here, this is just an ELISA where Tony collected conditioned media from trigeminal neurons cultured alone, or with the parental Merle tumor cells, or the exosome compromised cells. And what he found was that the trigeminal neurons release a low level of substance P. But if you culture them with the parental line, that gets boosted up quite significantly. But if the tumor cells can't release exosomes, that goes back down. So the exosomes are important here. When we looked at IL-6, here we saw that the tumor cells, they're expressing really low levels of IL-6. But when we stimulate them with substance P to sort of mimic what might be happening at the tumor bed with the tumor infiltrating neurons there, now they start releasing a lot more IL-6. And if we block the substance P receptor, which is NK1R, we can dampen this. So substance P is important. Then we asked, well, what about IL-6? When we culture the trigeminal neurons alone, they're releasing a decent amount of IL-6, The tumor cells really aren't. When you combine them, we can boost the IL-6 expression. And if we block the substance P receptor, this goes back down. So there is some sort of communication happening between the tumor cells and the neurons that's affecting the release of the amount of IL-6. But which one is releasing the most amount of IL-6? I'll be honest, I didn't know that neurons could release IL-6. That was a surprise to me. But when I dug in the literature, that wasn't new. That's been known. It was new to me. So we ended up getting an IL-6 knockout mouse. And we did this experiment just to convince ourselves that the neurons were actually the source of the IL-6. So when you co-culture the IL-6 knockout neurons with the tumor cells, there's no IL-6. If you co-culture it with the wild type DRG, so these can express IL-6 with the tumor cells, now we see IL-6 expression. And when you co-culture with the exosome compromised tumor cells, there is none. So the majority of the IL-6 is coming from the neurons, not the tumor cells. And we can show this in a dose-dependent manner. The more tumor cells that we culture the DRG with, the more IL-6 we get. Okay, so what about the immune cells? So IL-6, I said, has a big role in the recruiting immune cells, the function of the immune cells. So what's happening in the animal itself? So we implanted tumors into wild-type mice. We harvested the tumors and ran a very simple flow experiment just to get an idea of what's the immune composition. When we do this in a nerve ablated animal, we see a decrease in the number of myeloid-derived suppressor cells and a significant increase in the CD8 cells, suggesting that the tumor-infiltrating neurons are promoting an immune suppressive environment, which is what the MDSCs do. But is this unique to head and neck cancer? Is this unique even to just our tumor cell itself? So we went back to our very close collaborators in Canada, Sebastian Talbot and Miriam Ahmadi, who work on melanoma. And we said, let's just see if the same thing is happening in melanoma. So what Seb and Miriam did was they took their melanoma line and they grew them in nerve intact and ablated animals. They saw exactly what we and everybody else in the field see. If you ablate the neurons, the tumors will grow slower. But what about the influence on the immune response? They saw something very similar. When they grew the tumor cells, the melanoma cells, in nerve ablated animals, they saw a decrease in the MDSCs, just like we did in the head and neck tumors. But they went a step further and they said, okay, what is happening to these MDSCs under these conditions? And you don't even have to know what these transcripts are, but there's a huge difference. There's a complete shift in the transcriptome if the MDSCs were in the nerve wild type or nerve ablated animal. So the presence of the neurons is changing the MDSCs completely. So then we did this experiment. And I'm going to walk you through it because when Tony first presented this to me, my head was spinning. But it's actually a very simple experiment. So what he did was he said, all right, I'm going to culture the parental cancer cell line, the exosome compromised variant of it, either alone or in combination with DRG neurons or the DRG neurons alone. Okay, that's step one. He then took bone marrow cells from wild type animals and would then expose them to the conditioned media from these different conditions. So he's basically asking, do the tumor cells or the neurons or their combination release factors that can turn these bone marrow cells into MDSCs? Is the presence of the neurons together with the tumors inducing some sort of differentiation of MDSCs? So I'll walk you through this. So the positive control first is if you take MDSCs and expose them to IL-6 and GM-CSF, they will take on an MDSC-like profile, and that worked very well. If you don't expose them to those factors, they don't. So when Tony exposed them to conditioned media from the DRG alone, they don't really look much different than the negative control. But when he exposes them to conditioned media that came from the neurons co-cultured with the parental line, these look almost like our positive control. So there's something in that milieu that was changing the bone marrow cells into MDSC-like cells. But if we co-culture it if we expose the bone marrow cells to the co-cultured media from the exosome compromised tumor cells with the nerves, this goes back down. So again, exosomes are very important. And again, the ablated and compromised animal conditioned media look just like our negative control. So the combination of tumors and neurons release a bunch of things that can turn bone marrow cells into MDSCs. And when we looked at the ability of these conditions to induce migration, so this is just a simple transwell migration assay, again, it was the combination of DRG from wild type neurons and the wild type parental tumor cell that can induce migration of these MDSCs. And when we looked at markers of MDSCs, again, it was that combination that was leading to not only differentiation of bone marrow cells into MDSCs, but their migration as well. And so that was really our first clue that it's a sort of a cooperation between the neurons and the tumor cells to recruit MDSCs to the tumor bed and induce this immune suppressive environment. Okay, so when we looked at the CD8 cells, so that was MDSCs, what about the CD8s? We looked at markers of exhaustion, PD-1, LAG3, and TIM3. When we take splenocyte-derived CD8 cells and expose them to vehicle, they have a low level expression of these exhaustion markers. If they're exposed to conditioned media from neurons, this actually goes up. So the neurons alone can be exhausting the CD8 cells. If they're exposed to the purified exosomes from the tumor cells, nothing really happens, and the combination is not very impressive. So it's really a contribution of the neurons themselves that are exhausting the CD8 cells. And the same thing was true when we looked at other markers of exhaustion, interferon gamma and IL-6. So then we said, all right, that's really interesting, but are there changes occurring to the neurons, right? So we had just been looking at how the neurons are influencing the immune cells, but is this a bidirectional conversation? Are these immune cells or other components changing the neurons? So again, very simple study. We isolated and cultured neurons alone with splenocyte-derived CD8 T cells or the tumor purified exosomes or the combination. We took out the neurons and did RNA-seq. So what you can see is there were huge differences. If the neurons were co-cultured with splenocyte-derived CD8 T cells, we saw an increase in ATF3, which we had seen before. That's that transcription factor that's promoted when neurons are injured, as well as regeneration-associated genes, which would be expected, and IL-6. If they were instead exposed to the purified exosomes from the tumor cells, now we again see ATF3 go up, but now we see substance P and CGRP go down. This is actually what you'd expect to see. When neurons are undergoing injury repair, they actually downregulate their differentiation markers, and almost as if all the energy of that neuron is going towards repairing and regenerating those axons or those fibers, so they don't care about maintaining differentiation markers. So when you see high ATF3, you kind of expect to see low substance P and CGRP. And when they were exposed to the combination, again, we saw very similar things. ATF3, IL-6 go up, substance P and CGRP go down. Okay, so this is interesting at the transcriptional level. The neurons are undergoing changes, the immune cells are undergoing changes, but what are the functional consequences of this? So we wanted to see if there was a connection to what we're seeing in our patients, right? Because all this is in vitro, in vivo, and mouse models. We care about the patients. So we started working more closely with Caitlin Williamson and Chad Spanos. He's a head and neck cancer surgeon and has a lab at Sanford. And we collected blood from head and neck cancer patients, both males and females, and normal controls, and purified the exosomes from them. And we asked a simple question: do these exosomes have the ability to alter neuronal activity? And so we do this by exposing neurons, either trigeminal or DRG, they just have to be sensory, to the exosomes and looking at their calcium response to capsaicin, which is a TRPV1 agonist. And it was really interesting. So the control exosomes, they don't do a whole lot, But if the neurons were exposed to exosomes from cancer patients, head and neck cancer patients, we saw a robust calcium response to these neurons, suggesting that the exosomes were inducing some sort of change on these neurons that were making them hyper responsive to stimuli. And then we also noticed, the male samples are over here. Most of the female samples are down there. Is there a sex difference as well? We know there's a sex difference as far as incidence of head and neck cancer, but we wanted to know, is there something more on the biological neuroscience level that we're missing? So we started asking questions about differences between males and females. And we started really simply. This was work started by Sarah, who was a student in my lab, and then continued with Craig and Destiny. And she simply put tumor cells, and this time we switched to HPV negative, since these are the more densely innervated, and we implant them orthotopically in the oral cavity. She put them in males and females, and they grew slower in females, which was interesting. And we hadn't done this before, so we didn't know that was a thing, and it's highly reproducible. Many people in the lab have done this, and we see the same thing over and over again. Now, the female mice have a smaller body size, and we thought, well, it's smaller just because the body size is smaller. But when you normalize for body size, the tumor size stays smaller. We said, well, maybe this is something funky about this cell line. What about a different cell line? So we looked at the female variant of the HPV positive cell line. Female tumors were still smaller. And the same was true in melanoma. So it seemed that it was more of a general phenomenon that tumors grow slower in females, which is interesting. Then when we looked at the innervation, this is where it really became interesting. When we stain these tumors for beta-3 tubulin, which again is our pan-neuronal marker, and we quantify this, despite being smaller, the female tumors were more densely innervated. And this goes against what everyone in the field, myself included, have published, that the more nerves a tumor has, the faster it grows. But here now we have smaller tumors that have more nerves, which is completely counterintuitive, hard to understand. When we normalize for the size of the tumors, because they are smaller, they still remain more densely innervated versus the males. This was also true in the HPV positive line, and we missed getting significance just by a little bit here, but the trend was the same in melanoma. Okay, so what does all this mean? Where are these nerves coming from, and are they involved in a bigger circuit than we were aware of? So Jeff Barr in the lab, who's a cancer neuroscientist, staff scientist in my lab, he said, well, let's trace these. So the way we do this is we, again, orthotopically implant these cells. We let them grow, so they recruit the neurons, all these different things happen. And then Jeff will intratumorally inject a neural tracer. So these are fluorescently tagged markers. They could be, we use WGA, but there are many different flavors of these. And you let time go by, a few days. And what happens during that time is that the nerve terminals that are exposed to where you injected your tracer will take this tracer up, they'll retrogradely transport it, and it'll go back to wherever the neuronal soma is. And so when we harvest these tumors, we found that there were neurites positive in the tumor. We knew there were tumors there. And then there were also positive somas in the trigeminal ganglion, and specifically the V3 branch of the trigeminal. So we knew that these neurons were the ones that were infiltrating the tumor. And so that's where they were coming from. And when we compared the ipsilateral and contralateral side, it was very specific to the ipsilateral side. So there was no leakage to the other side in any way. And this, again, highly reproducible. Does this connect to a larger circuit? It's possible. So we looked at the brain. We let this tracer go a little bit longer, and we looked at the brain, and we found there was labeling in the spinal nucleus of the trigeminal, the parabrachial nucleus, and the central amygdala. This is a pre-existing circuit. The tumor didn't create the circuit. It just sort of hops onto it. And when we looked at control brains, because we know that circuit is there, when we first put 2 microliters of the WGA in that region, we didn't see any labeling, which didn't make any sense. We know that circuit is there. When we put more WGA there, we lit up the same circuit. And the idea here was that at the tumor, there is an increased density of nerves there because it's an active recruitment. So it takes less tracer to map this circuit than it would in the normal in the normal oral cavity. So this is the circuit that we identified. And when we looked at, and I'll remind you here, when we had looked at the ipsilateral and contralateral trigeminals, we saw these transcriptional changes. So this is in the periphery, the trigeminal ganglion. So do these neuronal changes now extend up to these projection areas that we know are connected to the tumor? And we looked at this by staining for two transcription factors, CFOS and delta FOSB. These are turned on when neurons have been recently active. And we compared these regions of the brain from mice that had tumors versus those that didn't. And we found that, yep, when there was a tumor, there was increased CFOS and delta FOSB in these brain areas, suggesting that possibly the changes that are occurring in the periphery of these neurons, the trigeminal neurons, was actually extending all the way up to the brain. What about glia? We saw a very similar thing. When we look at markers of glial activation, both for microglia and astrocytes, on the ipsilateral side in these brain regions, there was increased glial activation as well. So what does all this mean? If you have these changes in your brain, we start to wonder, well, maybe behavior changes. And why is this important? It's because cancer patients, no matter what kind of cancer you have an increased incidence of things like depression and anxiety. And while that might make sense, you just got a cancer diagnosis, who wouldn't be depressed? This is actually in long-term survivors. Patients 10 years or more, no evidence of disease, still have this higher incidence of mental health disorders. And that's never really been understood. And so we started wondering, maybe there was a connection here. So Austin Walls in my lab started doing behavior assays, which is weird for a cancer scientist to do. So we had to learn how to do this. And we initially looked at nesting, which is a measure of well-being. And we did the cookie test because our cages don't allow us to do 2 water bottles, so we couldn't do the sucrose preference test. So we did a cookie test, which I'll explain that in a second, how that works. So when we looked at nesting, if we genetically ablated the nerve, so now the tumors aren't getting as densely innervated, we can restore nesting behavior, not back to normal, but it's better than in the wild type. And in the cookie test, what you do is you put a certain amount of cookie, I think it was 1 gram, and what you measure is the time it takes the mouse to interact with that cookie. And interaction means taking that first bite. And so initially the mice don't know what a cookie is, but once they realize, oh, I really like this, mice like sugar a lot, that time to interact gets shorter and shorter and shorter. And so when we look at this, we do see the time to interact getting shorter until a point as the tumor is growing, and then it starts to level off and maybe go up a little bit, but not with the ablated mice. This time gets shorter and shorter and shorter. So even though they still have a tumor, their behavior is starting to get more restored compared to the wild type animal. When we do the genetic approach, there are complications with that. You're ablating all TRPV1 expressing neurons throughout development. And so maybe that's changing what we're seeing. So we wanted a more rigorous way to do this. And if you use this a potent analog of capsaicin called recipratoxin, or RTX. This will ablate TRPV1 expressing neurons in adults. So development's already happened normally, and now we can take those animals, expose them to RTX, and then ablate the neurons. And it's a very simple protocol to do. And we see that similar to the genetic ablation, when we chemically ablate the nerves, we can restore at least partially the nesting behavior. Okay, what about sex differences, right? I already showed you there's a difference in innervation. I'm showing you there's a difference, the innervation has an effect on behavior. Is there a sex difference in behavior as well? So we looked at the neural circuit in males and females. It was exactly the same. Nothing had changed. When we looked at behavior, there was absolutely no change. Nesting was the same. Here we looked at the open field test, exactly the same. And so really the change in between the sexes was local. This was happening really just at the tumor bed. Everything else looked very similar. What about treatment response, right? Because the radiation, at least, is given locally. We know there's a local difference in innervation. Maybe there's a difference in treatment response. So we gave tumors to male and female mice, and they got treatment. In this case, was cisplatin radiation, which is standard of care, and we tracked tumor growth. And as expected, when you treat tumors, either in females or in males, they grow slower. We know that it's going to do that. When we looked at innervation, though, this was actually more interesting. Post-treatment, so there's some residual disease, these tumors are actually more innervated than we started with, even though the tumors grew slower because of the treatment. Now they're more innervated, and the females had an even higher effect. We said, all right, we know there's more nerves. What are they doing? So we harvested these tumors and we did sections of them and we analyzed their activity by microelectrode array analysis, which really measures the electrical activity of the entire tumor slice. And what we found was that even though there were more nerves post-treatment, they were actually less active. And this was true in males and females. So they're there, but they're not doing their thing. We said, well, that's the whole slice. We're really interested in just the neuronal activity. So we pulled trigeminal ganglia from tumor-bearing treated or not animals and analyzed them by calcium activity, and we saw the same exact thing. Post-treatment, they're actually less active, even though they're greater in number. Okay, what about in the brain? We saw a similar thing. So post-treatment, we can see a decrease in these brain regions of CPhos and delta phosph. So it seems to go throughout the circuit that the tumor is connected to. What about glia? Same thing. Glial activation goes down with treatment. And behavior gets restored. So here, I'll show you in the females, the nesting behavior decreases, but we can restore it with treatment. And in males, actually, we can make it look like wild type, even though there's still a tumor there and it's more densely innervated. What about metastasis? Again, this goes to treatment response, which is what we are really interested in. And this was actually kind of cool. So the females, right off the bat, have more lung mets than the males in the absence of treatment. And that doesn't statistically change with treatment, but there's a trend to more. The males actually that respond well locally actually respond well with metastasis. The treatment decreases their metastasis. But the females don't. Even though we saw that their primary tumor shrunk, they actually have more mets. They have more nerves there. Maybe there's a connection there. We're not sure. When we looked at the patient data available for this, females actually do worse, and they do have more mets compared to males. So this is actually phenocopying that, and it's very interesting to try to understand what's happening there. What about therapy? So we understand the nerves are there. There's a change in innervation with standard of care treatment. Can we somehow target this, right? Like we're interested in clinical application to what we're learning. So we said, all right, well, maybe can we silence the nerves locally, right? Can we give some sort of pharmacological intervention that will silence these nerves, and maybe it will be similar to ablating them. We'll see a restoration of behavior. So we partnered with a company that had this drug called NTX 2009. This company got bought out, so we can't get our hands on this anymore, but this was a charged sodium channel blocker. And it was interesting to us because it was in a gel form. So we didn't have to inject anything, which was much easier for us to do and easier for the animals as well. And so we put this gel on and we said, okay, what happens? The tumors grew slower. So if we silence locally, we can decrease tumor growth. And the one behavior, this is before we started doing a lot of behavior studies. We just did the tail suspension test, which is typically used to test depression drugs. And the idea is you hang the mouse by the tail, which I know seems terrible, but people do this. We did it. And what you measure is how long it takes them to sort of give up struggling, trying to right themselves. Like they're just sort of this, I'm not going to fight this anymore. And the more they fight, the better their mental capacity is, basically. And we saw that pre-treatment, there was no difference between the groups, but post-treatment, the NTX treated animals they had a shorter immobile time, which suggested that the drug was actually affecting their despair in a good way. So unfortunately, when we tried to repeat this study, this was our pilot study, and we let the tumors grow a little bit longer, we lost this effect. And that's because penetration of the drug became a problem. The bigger the tumor grew, we weren't getting it deep enough. And so delivery is a challenge. right? Especially when you want to do something topical. So we said, all right, let's try something else. So Exparel, it's bupivacaine liposomes that are long acting. So we picked this because we didn't want to have to inject daily. This gave us at least 3 days. And we saw that Exparel, so this was a intratumoral injection. Exparel did decrease tumor growth. And what about behavior? We're looking at that now. So what we've learned so far, we're really still working on this, how to therapeutically target it. So exosomes are important for recruitment. They're essential for initiation of the tumor. The tumor infiltrating neurons are transcriptionally modified. They take on this transcriptional injury profile. We see that the combination of the neurons and the tumor cells can modify the milieu of the tumor microenvironment. They cooperate in recruiting and differentiating myeloid-derived suppressor cells and exhausting CD8 T cells, so they induce this immune suppressive environment. And the transcriptome is changed on both sides, right? It's a bidirectional communication. Peripheral tumors are connected to a circuit, right? So we know there's an effect on the brain, which this was actually really interesting for the field, that it goes through protection areas in the brain and can affect behavior. There is activation of neurons and glial cells in the brain, and it impacts behavior. When we do standard of care treatment, we can attenuate these changes. We can restore behavior. In the females, we did not restore the running wheel. So not all behaviors, but some. But the females kind of turn everything upside down, right? The female tumors are smaller, but they're more innervated. They have increased innervation post-treatment. They're less sensitive to agonists. They have more metastasis in the lungs compared to males pre and post-treatment. So we need to start understanding this more, because right now, when patients come into the clinic and they talk to their oncologist, males and females are treated the same, at least for head and neck cancer. But we're showing you the disease is different, and they're responding differently to treatment as well. So we need to understand this better. So what are the molecular mechanisms leading to these sex differences? We really don't know now. That's where the lab is going. What's generating the difference in the tumor microenvironment, the neuronal sensitization, the altered immune response? And does this happen in other cancers? So we actually did look at ovarian cancer, and post-treatment, and these are patient samples, not mice, there's actually also more innervation post-treatment in high-grade serous ovarian carcinoma. So this may be a general phenomenon. And how does this inform on treatment resistance and metastasis? That's what we're really interested in, because that's what our patients are dying from. So I'll leave it at that. These are my funding sources. I tried to highlight the people who did the work and all my collaborators. And thank you again for the opportunity to come here and share our work. And I'll take any questions. Yeah.
So, hi, I'm Sam Dudley from the University of Minnesota. I'm a cardiologist, so I'm confused.
Okay.
I think what you told me is that neurons infiltrating cancers, or at least head and neck cancer, increase the immunosuppressive environment by depleting CD8 cells and perhaps increasing myeloid tumor suppressor cells. And yet you showed an elevation in IL-6, which would be actually an increased inflammatory response, or at least one measured increased inflammatory response. And in general, in the cancer literature, you see both immunosuppression causing cancer and increased immune activity causing cancer. So that generally confuses somebody like me when you say both things good. And you also showed that neurons were not always correlated with increased cancer, especially in the women, where you have
increased growth,
cancer.
Yeah.
So I'm confused. Is it not enough immune response? Is it too much immune response? Or is there an intermediary between neurons and cancer that we're missing? And also, could you use neurostimulation to modulate cancer?
Okay, yes, we're actually trying to do that now so that you could use optogen. So neuroscientists are really clever people, and they have designed all these really cool tools that now cancer neuroscientists are taking and utilizing to study cancer. So you can activate and inhibit neurons using optogenetics or chemogenetics. And we're trying to do that now to ask that question. If we can modulate the activity either locally or maybe a little bit more further away, is that going to change the immune response, the inflammation response in the tumor, and what's that going to do to tumor growth, and how are males and females different? So I don't have a great answer because we don't have all the pieces yet. It is confusing, you're right. And I think part of that is because we don't fully understand the molecular conversations that are happening. And they're not just bidirectional, they're tridirectionals and quadridirectionals. There are a lot of things happening. And I didn't talk about this, the tumor infiltrating neurons are also affecting cancer-associated fibroblasts. It's not just the CD8 T cells or the MDSCs or the tumor cells themselves. The tumors release substance P that directly induces proliferation of the tumor cells. The neurons. The neurons are releasing CGRP that in another way induces exhaustion of the CD8 T cells. So CD8 T cells express RAMP1, which is a CGRP receptor. And when that engagement happens in the tumor bed, that also induces exhaustion of the T cells. So there are a lot of things happening there, and it is confusing. And that's why we're trying to go back to tissue culture and ask more simplified questions before we go back into the animal and ask as a whole what is happening. But the field is growing, and I think we're going to start to get these answers.
Does it matter which neurotransmitter?
It does. So not all tumors are infiltrated by sensory neurons. Some are infiltrated by sympathetic, parasympathetic, or both. And what we know about that is the parasympathetic innervation is pro-tumor, so inducing increased tumor growth, increased suppression. But parasympathetic seems to be putting the brakes on that. And they work during different stages of tumor initiation, whereas the sympathetic is more early, the parasympathetic is more sort of late at the metastatic stage. So because the recruitment is induced by the tumor, remember, they release all these factors that recruit local regional nerves. The type of neurons that are, the type of nerves that infiltrate tumors completely depend on where the tumor is. So if we've actually taken our oral tumors and put them in the hind limb to see what happens, they get innervated by lumbar DRG. So it's totally regional.
Be an interesting project to look at parasympathetic tone and the occurrence of cancer. It's an easy epidemiologic project. You can measure parasympathetic tone to heart rate variability. It's also good.
That would be very interesting.
It's known that people with higher heart rate variability or higher vagus tone live longer. But the question for me is why?
Yeah. Yeah. I mean, there was a study that was Hubert Hundermark, I think, did this, where he looked at patients that had spinal cord injury, men, spinal cord injury or controls. And if they had a spinal cord injury, their incidence of prostate cancer was less because they couldn't get them innervated. So the nerves are really, really important. Yeah.
Speaking of local environments, but the brain itself, are there any similarities and differences between the role of nerves, innervation, and tumor growth, like gliomas or metastases in the brain as opposed to implant?
Yeah. So Michelle Manji does a lot of that work, and she's been looking at gliomas. This is actually really interesting. So the glioma cells actually form synapse-like structures with normal neurons. And she identified, I think it's called Netrin 3. I'm not sure on the name, don't quote me on that. But there is a direct molecular communication where the normal neurons are actually inducing increased growth of the glioma. So they're intricately connected to each other, and the communication is active. Like it happens early, it continues on late. She can block this with receptor blockade. So there are therapies that she's looking into now to see if we silence this communication, can we get a control of the glioma growth? And it seems to be working at least in animals. So it happens in the brain too.
That's fascinating. I'm wondering if there are any opportunities to seek support if you're interested in this line of work from Brain Initiative, John Nye, they're interested in circuits, connections, different types of cells in the brain, how they interact in health and disease. And seems like a great topic.
Yeah, so as far as I'm aware, we're the only ones that have really looked at the connection of a peripheral malignancy to the brain. Michelle does all the brain stuff. Her and Frank Winkler are huge in the brain part of it. And we're starting to get more people interested in the connection between the peripheral tumors and the brain, but that's a great suggestion.
Hey, this is wonderful stuff, and it's stimulated some sort of latent neurons somewhere back in my brain. I used to be a psychologist and frozen and everything else. And there's this old literature which I often looked down on that looked at the incidence of cancer among schizophrenic patients. And it's lower. It's robustly lower. And I always attributed that to, well, you're just not working these people up. That is, there's latent cancer there, but you haven't seen it. But it's even true for lung cancer. And certainly years ago when everyone smoked on chronic wards, you would have expected an enormous increase in lung cancer among chronic schizophrenics, and you don't see it.
What do they get treated with for schizophrenia?
Oh my gosh.
I have no idea. I
That's
ask.
the question.
Anti-dopaminergic agents.
So I'm sure that plays a role. So they've done epidemiological studies, and I think it was in breast cancer. Women that happened to have been on propranolol had lower incidence of breast cancer. And if they had breast cancer, it was more luminal A, luminal B, not the baddies, right? And so there's a great amount of interest to look at if you're taking drugs for a completely different reason that are interfering with neuronal function, how is that affecting the tumor? And it is. So beta blockers are now being tested. I think it's a phase 2 clinical trial to see if they treat, and it's done in the context of, I believe it's breast cancer, but just perioperatively. There have been studies showing that post-surgery, there's women that had surgery had an increased metastatic rate. And so they're treating these patients with propranolol pre and slightly post-surgery to see, can we now affect their metastatic rate? And so far it's looking like it does. So the idea is to take that information once the trial is done, and maybe we start using something like propranolol as standard of care.
I wonder about the sex difference. This is so obvious. I'm sure you've probably thought about it. Do castrated males look like females?
Yeah, we haven't done that yet. We've thought about it. I don't know. This is
That's an easy experiment.
yes, very easy to do. This is the first study looking at sex differences in innervation. I'm sure we're going to see others coming out in other tumor types, and I expect we'll see something similar. So we need to do those kinds of studies, and I still don't understand how it's happening, right? How are they recruiting more? They're not releasing more exosomes. They're not releasing more neurotrophic factors. The tumor line is exactly the same, but in the host, the response by the host is very different. It's fascinating. Okay. All right. Thank you very much.